Synthetic theory of evolution. So, with the development of knowledge in molecular level, molecular level knowledge in evolution, like the knowledge about DNA, RNA, genes, genome, etc. There was a further synthesis of neo-Darwinism. Further knowledge from paleontology, systematics were also added to this, and there was a new theory, or the neo-Darwinism got further synthesized, which we call it as the new or modern synthetic theory of evolution. This modern synthetic theory of evolution, right, was propounded by a number of scientists, most notably Dobzhansky, Fisher, Haldane, Erwin Mayer, G.L. Stebbins, etc. In fact, it was G.L. Stebbins in his very famous book, Process of Organic Evolution. Process of Organic Evolution. It is in this book, right? He proposed the synthetic theory of evolution. Synthetic theory of evolution. The synthetic theory, right? This again includes five very important factors. Five important factors in evolution. This is number one. Gene mutation. Gene mutation. Then number two, variation and variation by recombination. I will explain you what is the variation and this recombination. Then heredity. Fourth one is natural selection. And fifth one, isolation. Natural selection and then isolation. So further, right, there are some three accessory factors in synthetic theory of evolution. Apart from the five major factors, there are three accessory factors in synthetic theory of evolution, which also plays a major role in evolution. Please try to make a very important note on these three accessory factors. These are number one, migration of individuals. Migration of individuals from one population to another population. Okay, so this is also a very important source in evolution. So by migration, right, one person from a population may carry his own genes and he may make others to practice. For example, we Indians, right, we are migrating to Europe. We are migrating to North America and we carry our genes with us. Dowry system is very much in our genes. And when we practice dowry system, right, they will also think, hey, how nice these guys, just because they got into some employment, right? How much money they are extracting from the girls? Why can't we also try that? So that will make even the jealous and greed people there to follow our customs, right? So migration of population is also a very important access. Here they say it is an accessory source here for evolution. First one, migration. Number two, hybridization between races. That is breeding among different population. That will also bring in a number of new genetic combination. If a Mongoloid people, if they breed with a Negroid population, there may be some exchange of genes. The extraordinary or the quick reflexes which the Mongoloid people have, right? It is because of that quick reflexes only Mongoloid people excel in few games like badminton, table tennis, etc. Have you ever seen a Negro playing a table tennis? The reflexes are relatively poor. But Mongoloid people, right, they have very quick reflexes. So, hybridization between races, not just in human population, among other population also, may also bring in a new combination in genes. Then the third one, effects of chance acting on small population. Chance, right, this is also a very important source in evolution. See our Salman Khan, what a muscle he has, and what a good actor he is. But if he continue to remain without getting married, then there are no chance for the population to get his genes. His genes may end with that. So once when by chance if he gets married, so there is a chance for the genes from him to penetrate into the population. So, but there are also chances that these genes may not penetrate. So chance also plays a major role in making genetic combinations. So these three are the accessory factors which were not discussed earlier in other, be it Darwin's theory or neo-Darwinism. Migration of the population, migration of people from one population to other, hybridization between different population, and the chance acting on a small population. So now let us discuss the other aspects of the synthetic theory of evolution. First thing is mutation. Now, because of the COVID and the first wave, second wave, third wave, guys, you all will be familiar with the term mutation. They keep saying mutant strains, mutant strains, mutant strains. So, what do you mean by mutation? Mutation, right, refers to a sudden or spontaneous change in a sudden change in genetic makeup of an organism. This you might have studied during your school days. So it may happen due to a variety of reasons. Few are known to us and many are not known to us. For example, if we are exposed to a nuclear radiation, alpha, beta, gamma rays, suddenly, right, that may affect our genetic system and that may induce some mutation. There are also some chemicals which creates mutation. The most important chemical is ethyl methyl sulfonate. We call it as EMS. Ethyl methyl sulfonate. So if you are exposed to this chemical, right, even this is capable of creating mutation. So mutation means this is our genetic makeup. If this C, right, if this is this gene is responsible for curly hairs. Suddenly there may be a mutation here. The C may get converted into lambda. As a result, this won't express in that generation. But the kids born to him, right, because he'll be having this type of genotype, He may have kinky and woolly hairs, like the one found in Negroes. So mutation refers to a sudden change in the genetic makeup of an individual, and there are two types of mutation, may be positive and negative. So mutation tend to produce changes, but in subsequent generation, right, reverse mutation may happen, and which may bring it back, it may bring back the genome to normal. Reverse mutation also happens in the population to bring back the changes into the original condition. But still, sometimes forward mutation may happen, or there are two things. Forward mutation brings in changes. Reverse mutation may restore the population to its original stage. But sometimes the forward mutation may happen at a greater rate, so that the changes will be prominent and changes may get perpetuated. Changes may filter into the population. Okay. And most of the genes produced by the mutation are recessive. Most of the genes produced by mutation are recessive. Now, let me introduce you to the concept of recessive genes and dominant genes. Homozygosity and heterozygosity. We all know that Gregor John Mendel, he was a great Austrian old monk. He's an old Austrian monk or Austrian old monk, whatever may be the thing. He had a lot of free time, and during his free time, he did some experiment with the Pisum sativum plant, pea plant. He did some experiment with the pea plant, Pisum sativum. So what he did, he selected tall plants, and he crossed that with the dwarf plants. In the first generation, right, he managed to find only tall plant, and he couldn't find this is F1, filial one. He managed to find only tall plant. There was total absence of dwarf plant. It was a big surprise. How it happened? Then what he did, again he selected seeds from F1. In the F2 generation, right, he managed to find a few dwarf plants. There were a combination of tall and dwarf plants. So in one generation, dwarf plant went missing, and it reoccurred in the next generation. It was a matter of surprise for him. So what he did, he repeated the experiment again and again. And in every single time when he repeated the experiment, he found that in the first generation, the dwarf plant went missing, and in the second generation, dwarf plant reoccurred at a definite proportion, at the ratio of 3:1 roughly. This made him to come to one conclusion, that there should be two alleles. He made a number of mathematical calculations to find that there should be two alleles responsible for one gene, and these alleles should be capable of segregating during gamete formation. He was far ahead of his time, and I don't know what mathematical models he used to come to this conclusion. Nobody at the time was ready to even accept his theory, his hypothesis, which was later rediscovered in the first half or the first few decades of the 20th century. And they found what a genius he was. He found that one allele comes from the father side, and one allele comes from the mother side. So if it is a tall plant, T. This is one allele, this is one allele. One come from the father side, another come from the mother side, and this makes one gene. And once again, when this individual, right, when he is going to give birth, there will be a gamete formation. In human beings, it is the formation of sperms in male and ovum in the female. So during gamete formation, again these two alleles, right, segregates. So in one gamete, we will find only one allele. So that this will pair up with that one allele from the mother side. Again, the 2n number will be restored. This he managed to identify, that is, two alleles were responsible for one genes. So he did the experiment like this. See, he selected one tall plant, one dwarf plant. So during gamete formation, the two alleles, right, the 2n number will get reduced into n. It happens for human beings also. In sperms or in ovum, we cannot find the 46 number of chromosomes. We can find only 23 number of chromosomes. This 23 number of chromosomes from the sperms will fuse with the another 23 from the ovum, and finally the 2n number 2n is equal to 46 will be restored from this zygote into that new individual. So during gamete formation, these alleles segregate. When he crossed it, right, one allele managed to mix with one and he got this plant. And this is also a tall plant. Also a tall plant. See, this is also a tall plant. This is also a tall plant. But what is the difference here? There are two things called as phenotype and genotype. Phenotype means a physical expression, physically how one look, that is phenotype. Genotype means a genetical makeup of an individual which makes him look like that. That is genotype. Okay. This plant is also tall. This is also tall. So phenotypically, both the plants are tall, but genotypically, both the plants are not the same plant. One is, this is homozygous tall. And this is heterozygous tall. So are you okay with the term homozygosity and heterozygosity? In homozygous condition, both the alleles will be same. On the other hand, under heterozygous condition, both the alleles may not be same. There will be two alleles, but both the alleles will not express here. Only one gene will express here. So it is this big T which is going to express, and the small one, right, will not express here. This gene suppresses the expression of this particular gene. So this is a dominant gene and this is a recessive gene. This is a recessive gene. So under heterozygous condition, it is only the dominant gene which will express. This is called as law of dominance. Then let us now continue our Mendel's experiment. In the next what he did, he collected seeds from the F1. Here one plant is, both the plants, right, have this genotype. During gamete formation, this T will segregate. As a result, he got a discrete combination, big T, small t. When he crossed it, there is a random chance for all the genes to combine. So the chances are T big T, T small t, T small t, small t, small t. So here the phenotypic ratio is 3 is to 1. 3 tall and 1 dwarf. But genotypically they all are not same. This one is homozygous tall. These two are heterozygous tall. This is a dwarf plant. This is a dwarf plant. Okay. So recessive genes express only under homozygous condition. Only when dominant gene is not there, right? Recessive genes can express. And see, in the last generation, where from the small t came? It came from the parents, right? It is because the small t paired with this large T for one full generation, it will not inherit any character from the tall gene. And when it segregates, it has its own character which it will express. This is called as law of independent assortment. Law of independent assortment. Guys, now are you okay with the terms like what do you mean by homozygous? What do you mean by heterozygous? A dominant gene, a recessive gene. So how the allele, what do you mean by a allele? And how the alleles segregate during gamete formation. Okay, so do not worry. Once again, right, I'll be repeating this soon when we discuss Hardy-Weinberg law of equilibrium. So that even if you do not know anything in science, I will make you to understand. Further, one more time, Kavita madam will also trouble you in teaching all this homozygosity, heterozygosity, alleles, dominant gene, recessive gene. So now come back to our synthetic theory of evolution. So most of the genes produced by evolution, right? So most of the genes produced by mutation, the genes will be recessive. The recessive genes, right, may continue in a population without getting expressed. Without getting expressed. And one and only, right, when it pairs up with a similar recessive gene, it will express. This particular thing, right, from starting till end, we'll discuss in anthropology. So even if you're not able to understand anything, right, with the passage of time, you will grab it because we'll be discussing n number of examples about homozygosity, heterozygosity, dominant gene, recessive gene, etc. Okay. So most of the genes, right, are produced by mutation are recessive genes, and they express phenotypically only under homozygous condition. So mutation, right, produces lot of variation, and this variation, right, see, for example, if there is one disease called as a sickle cell anemia. The sickle cell anemia, right? This disease is common among the people living in Central Africa, in Congo, this region, among the black Negroid population. Here, for them, the hemoglobin, right, instead of being round, it will be sickle shaped. Only this portion of the hemoglobin, right, will be active, and they cannot survive for a long time. Suppose this is genetic again. This is a homozygous recessive disease with, or suppose this is a normal cell, and if this is responsible for sickle cell anemia. Okay, some mutation may produce the gene, but most of the genes are recessive. Under recessive condition, the disease will not express. Only the dominant gene will be expressing. So in the population, right, silently there may be n number of carriers. And suppose if there is a marriage between these two, if there is a combination like this, the individual will be normal. But only right when not these two genes combines, that gene will express. Okay? Okay, then fine. So gene mutation tend to produce variation in a population. Just what we remember here is mutation is capable of producing changes, and most of the genes produced by mutation are recessive genes, and these genes will express only under homozygous condition. So to make you understand the term like homozygosity, heterozygosity only, I just explained you all the things. Guys, hope you are comfortable with the terms like homozygosity and heterozygosity, genotype, phenotype. Okay. So next is the variation and the recombination. So the variation, right? The variation happens due to a number of reasons. So it is because of genetic combinations. So I told you here, right? Every individual has its own genetic makeup, and when they breed among themselves, right, different genotypic combination may happen. So, for example, earlier in Mendel's experiment, there was only one genotype, and there was one more plant with another genotype. As there was interbreeding among them, right, we managed to produce one more genotype. Variation. So variation may happen because of the combination of genes. Combination of genes. It may also happen because of a random mixing of chromosomes between the parents. And even not during meiotic division, during gamete formation also, too many things happens actually. During the meiotic division, many things like crossing over may happen. For example, this is one chromosome. There is one more chromosome lying nearby. Sometimes what happens, these arms may overlap. As a result, this portion of the chromosome may come here and this portion may go there when things get when the cells divide. This we call it as crossing over. There's not to worry much about this thing. You can simply say variations are there. Variations happens because of a number of genetic recombination. Number of genetic recombination. That is sufficient. Next is a heredity that we know. Heredity. So, it is our genes which are responsible for heredity. We inherit most of the characters because of the genes. So, in order that these variations produced in an organism should have evolutionary significance, these changes should be hereditary. It should be there in the genes. Then finally, natural selection and about isolation. I think about natural selection, there is no need for me to explain you again. But how isolation leads to evolution? Isolation plays a major role in changing the genetic makeup of an organism. I'll tell you one example. Earlier we have discussed that there are many scientists who were instrumental in bringing this theory, synthetic theory of evolution. One such person is Sewall Wright. And he proposed a very big hypothesis. Seawall right effect, which is also called as a genetic drift, about which we will discuss in length soon. For genetic drift, right, he gave one number of examples. That means few genes without having any advantage to a population, right, it may circulate in that population, it may get fixed. When Napoleon was arrested after the Napoleonic Wars, he was imprisoned. This looks like Africa, right? Saint Helena. This group of islands are also called as Bread Bay. Napoleon was imprisoned in Saint Helena Islands. That too, in particular, one island called as no. There are some group of islands. He was in a island called as Elba, and the group of islands right here is called as Tristan. Tristan Island. Okay, so it is from Elba when he was released, right? Napoleon Bonaparte gave the very famous palindrome. I hope guys you know what do you mean by palindrome. Malayalam is a palindrome. Liril is a palindrome. That means if you reverse it also, right? Liril, the same thing will come. The very famous palindrome which Napoleon said was able was I ever I saw Elba. Able was I ever I saw Elba. We can reverse this from the end. ABLEVWAS I ever I saw Elba. There is one very famous palindrome about Panama Canal. A man, a plan, a canal, Panama. Note it down. A man, a plan, a canal, Panama. Okay, so leave all these things. So Napoleon was imprisoned in Tristan Island in Africa, here in the Atlantic. To secure his arrest here, right, one garrison was moved from Europe. Maybe some 10,000 or 20,000 soldiers were moved here to Tristan. After some time, right, Napoleon was released. After his release, right, there is no need for these soldiers to stay here in the remote island. So most of them preferred to return back to Europe. But a few handful of soldiers with their related families, maybe not more than 10 or 20 families here, they preferred to stay back here. Why to go back there? If you go back there, right, my brother will create issue, my neighbors will create issue, or I need to fight for the property. How peaceful these islands are. That too, most of the people are vacating. Nobody will be there to question us. Let us stay back here in the island. And few group of people, right, they stayed there in Tristan itself. So when this huge population moved here, right, from Europe to Tristan, in that huge population, one or two, right, had a disease called as retinitis pigmentosa. Here, right, because of this disease, right, the retina, the white portion of the eye, right, retina means the white portion of the eye. The retina becomes red in color, or you can see number of red patches in the retina. Number of red patches in the retina. See, one in a million here had the disease, but when this 10,000 people moved here, right, one managed to carry that genes. And when everyone left, right, in that 20 families who remained there, that person managed to stay here with the genes. So when this population, right, they were breeding among themselves, because there is no scope for them to interact with the people in Africa or Europe. They were completely under isolation. When these isolated people, when they continue to breed among themselves, that gene for the retinitis pigmentosa, right? It multiplied among them, and that gene become very prominent. Now, 9 out or 8 out of 10 will be having that red color eyes. This is one example to show that isolation can produce variation in a population. Here, genes are responsible for the variation, but here, how the gene got multiplied and how it become prominent? Isolation. So, isolation may alter the genetic makeup of the population. Here, from neo-Darwinism, individuals, the basic unit of evolution is not an individual but a population. So, whatever changes we are going to, whatever changes we are going to discuss here, right? That changes we will be discussing in terms of population. So, now this isolation and inbreeding brings in a change in the genetic makeup of the population. Over time, if they continue to live, they may altogether become a different population. So this may happen among a number of organisms. So one organism may look progressively dissimilar from the other organism because of isolation and inbreeding. This isolation, right, it may be psychological as well as geographical. This isolation may be or it may be geographical. Psychological means if a group of people, right, they may think they are superior and they do not want to mix with others, and they will continue to isolate themselves though they are in the middle of a large population, and they may continue to breed among themselves. This is psychological isolation. So geographical isolation, right, this may be due to mountains, rivers, deserts, ocean, etc., forest, etc. Okay. So keep in mind, isolation also plays a very important role in the change in genetic makeup of organism. This is what synthetic theory of evolution wants to explain. So guys, this is sufficient about, so further right, all the 5 factors may lead to produce a different population. And with the passage of time, the differences may become so prominent, and we can call this as a different species. We may call them as a different species. A different species. Okay. So let us stop with this today. And in the next lecture, or let us finish this small topic alone. There is something called as microevolution and macroevolution. Just note it down in a bit of paper. So microevolution means, I didn't mention this in your note because this is not so a prominent a thing, but sometimes, right, they may ask in our examination. So microevolution means the changes produced by the evolution are so small. And changes are not so prominent. Changes are not so prominent. So what happens? The changes are not so prominent. Hence, the organism may not look so different from its ancestor, and we cannot call it as a different species. On the other hand, macroevolution means the changes produced by the evolution is very prominent, and we cannot identify that from which organism this organism got evolved. We cannot even identify its ancestor, and now it is fit to call this particular organism as a different species. That is macroevolution. Microevolution and macroevolution. So guys, if you can understand even 50% of what I discussed today, that is sufficient, because continuously we'll be discussing about this gene, genotype, gene frequency, and I will make it very easy for you. So do not worry. Homozygosity, heterozygosity, dominant gene, recessive gene, okay, genetic combinations, etc. but it will not be very tough. This is the maximum limit. Okay, so have a great time, and let us discuss the next topic in the next class. So until then, have a great day. Thank you.